Yeah, this is really kind of surprising finding when we got the results because we all know that TP53 mutation is very common in triple negative breast cancer based on the previous data in the European Americans, so in Asian women, it's about like 80%. So it's already very high. But then we found out almost all of them have uh, the cases in our study in African American women have P53 mutation. We thought, oh, this might be interesting. It could be because we are using somewhat different sequencing technology than previous studies, because now we are using a longer read than previously. We're using 150 BP instead of 100 BP. So that might explain some of the mutations we can find in our data because we do find some more longer insertion deletion. So those will benefit from having a longer read sequencing. But it could also be because of the bioinformatic process we are using, the pipeline we are using to call those mutations. So we use four callers and to integrate all those results together. And then we also did very stringent, meticulous manual review to make sure our mutation calls are robust. And we even did targeted amplicon sequencing to validate our P53 mutation. So we are very confident that the findings of the higher than expected P53 mutation is real. So then that begs the question whether this is only in African American women or this is because less mutation were found in previous studies because somehow those are missed. So we are not entirely sure. So it could be technological, could be methodological, or could be biological. I think it still need to be seen. I don't have a clear answer to that, but certainly this will be a very interesting question going forward. Yeah, that's a good question. So HRD in triple negative breast cancer has been known for a while, and then has also been used to design trials of novel treatment such as like PARP inhibitors, and then patients tend to have a good response to those treatment if they are HRD high in those tumors. But then what is surprising to us, or what is novel in our finding, is the aging-related TNBC. Because from our analysis, we can clearly see there those two processes are going, and one is more dominant in some tumors, and then the other is dominant in other tumors. So since we have known more about HRD related TNBC, I don't think we know a whole lot about aging related TNBC, especially TNBC is usually linked to a younger women early age diagnosis. So in older women, I think what's driving that and then how to treat those, whether they will respond the similarly way to the PARP inhibitors or even to immune checkpoint inhibitors. I think that's still an open question. I think that has not been well studied. So I think going forward, more research might be needed in this area. Yeah, I probably would like to clarify that CD274 is not related with HER2 or HER2. That's PD-L1. So that's more related with the immune checkpoint inhibitor treatment. But for the HER2 amplification, this is a different task than what is normally done clinically, which is usually IHC or with or without fish. So testing protein expression. And in our analysis, we are testing the copy number changes at the DNA level. So There are certainly some inconsistencies between those two technology, and that's also known in the literature. We talk about this in the paper that this is not something we just found. This is also reported before. So I think currently the clinical standard method is to do the IHC and FISH for RB2 and for HER2 expression. So whether that need to be changed or not, I think that's a question that's beyond the scope of our study right now. Sure. Yeah. So, yeah, I think the major finding of our study is really that the mutational landscape of triple negative breast cancer in African American women is very similar to that in in European American or Asian women. So, whether that's tells us the causes are the same or not, or why African American women still have the highest incidence of TNBC. I think there could be two factors we need to look further. One is the actually we are in the process looking at genetic factors. So when I say genetic factors here, I mean the germline genetics, like the genetic variations we inherit, the especially related with African ancestral background. And there are evidence showing that African ancestry is actually related with higher risk of triple negative breast cancer. So we are doing a large study trying to find genetic variants specific to African ancestral background that could be related with TNBC risk. But then the other factor I think is less well studied, less understood is what kind of non-genetic factors that may be important in driving TNBC disparities. So especially now our study is showing that the aging related with TNBC, and that's actually related with obesity. So whether obesity is a contributing factor to TNBC, I think there's still more epidemiological studies are needed. But if that's true, considering that obesity is more prevalent among African American women, I think that could be a contributing factor to the disparity. But still, certainly, that need more research for sure. Yeah, that's really expert question. Uh, the mutation signature has been a fairly new development. I think maybe in about 10 years now. It was since it first developed. It's a mathematical extraction of the mutation patterns we can now get from the sequencing, the massive sequencing data. And there has been a lot of new development, very fast evolving field, and then the findings are certainly not the most consistent I would like to see. Actually, in our own analysis, we have tried several different approaches, trying to get the most consistent result. Sometimes you got mutational signature that just does not make sense. For example, if you see a mutational signature in breast cancer that is related with UV exposure, so oh, okay, so that may not be good. So I think some common sense is certainly needed. And then there's a cosmic database that people usually use and reference to compare the mutational signatures you find with those reference signatures to see whether those are consistent and also comparing your findings with the literature. Also, we did a lot of that. We went through all the previous TNBC mutational signature analysis to make sure our findings are not so different from previous studies. Sometimes they got excited about those novel signatures related with mutational processes, but after looking more into it, probably doesn't make sense sometimes. So certainly a lot of careful literature review and interpretation of your results is necessary. So I appreciate that question a lot. Thank you.